Dear viewers, welcome to online courses. In this segment, we will try to know about economic importance of viruses. Viruses are infectious particles that cause diseases in plants, animals, human beings, fungi and even lower organisms. They have molecular level of organization and are believed to be of negative importance for human beings. Human efforts have made viruses useful in many ways. They include vaccine production, medicine production, genetic engineering, pesticide formation, and others. First, role of viruses in vaccine production. Virus-based vaccine production is one of the major industrial endeavors related to medicine. These days, such vaccine production is a routine affair in most countries. It involves production of specific viral strains pathogenic for a particular disease under controlled conditions. Suitable culture media are developed for the purpose. In the earlier days, laboratory scale operations were more prevalent. However, gradually, continuous techniques utilizing more sophisticated fermentation technology have replaced earlier modes. After production of virus particles, they are separated from residual debris in the media by utilizing techniques like graded ultrafiltration. Viral vaccines confer immunity against infection by pathogenic strains of the same virus. The initial research on vaccines by Edward Jenner in 1796 started with a virus. Jenner used the cowpox virus, now known as vaccinia virus, as a live vaccine for preventing smallpox. The conventional vaccines synthesized using live attenuated viruses or killed viruses are easy to produce and economic, but have a number of disadvantages. Reversion of live attenuated viral vaccine to the initial virulent form has also been observed in some vaccinated subjects like live polio vaccines. Besides, live vaccines might also contain certain undesirable, undefined components as they are usually produced in animal cells. In addition, some of the viral vaccines are difficult to produce because they do not grow very well in culture medium. Hepatitis B virus is the best example. The inactivated viral vaccines are ineffective in some diseases. Thus, live attenuated vaccines serve as a best option. Besides inactivated and live attenuated viral vaccines, recombinant vector vaccines are nowadays being made for prevention of a number of viral infections. The genes coding for the major antigenic proteins of a virus are isolated and inserted into a vector, which can either be an avirulent virus or a bacterium. This vector is inserted into human host, where the vector replicates and expresses the protein products of the recombinant gene, generating either humoral or cell-mediated immune response. Viruses commonly used as vectors include vaccinia virus, adenovirus, canary pox virus, and attenuated poliovirus. Out of these viruses, vaccinia virus is the most commonly used vector. Vaccinia virus has a broad host range and remains stable for a very long time period. However, the main drawback with the virus is its large size and lack of unique restriction sites. Thus, foreign genes can be inserted only by homologous recombination. A number of genes coding for major antigen of pathogenic viruses and even bacteria have been inserted into vaccinia virus DNA and are allowed to express in animal cell culture. The important viral antigens include hepatitis B surface antigen, influenza virus, hemagglutinin proteins, rabies virus, G protein, and herpes simplex virus glycoproteins. It is also possible to simultaneously clone genes for 2 to 3 viral antigens in a given vaccinia virus, thus opening the possibility of vaccination with multiple antigens at one go. Recently, viruses have also been used to produce DNA vaccines. However, DNA vaccines are still in infancy and research is underway. In a DNA vaccination, a plasmid preparation with genes of pathogenic virus and a strong viral promoter are injected into the muscles of human host. The antigenic proteins of the pathogens are now produced by the muscle cell, evoking both humoral and cell-mediated immune response. DNA vaccines against AIDS, viral vaccines, herpes, hepatitis B virus, etc. are now being synthesized and tested. The second role of viruses is in gene therapy. The introduction of functional genes into human cells to correct defective genes by replacing them is known as gene therapy. Initially, gene therapy was used for the treatment of patients with genetic disorders. However, the application of the therapy have increased widely now. Viruses prove helpful in gene therapy as carriers of desired genes. Gene therapy is effective in cases where introduction of either a single gene or limited number of genes is required to correct a given disorder. The other important requirements of gene therapy include availability of a functional copy of defective genes known as therapeutic gene and a method of gene delivery. Gene therapy is largely used in cancer treatment. However, lately it is also being used in the treatment of some infectious diseases. An overview of gene therapy strategies is given in the figure. The other important role played by viruses is germline gene therapy. It includes the introduction of therapeutic genes into germ cells with the aim to correct defects in next generation. Germline gene therapy is currently not being used in humans and is restricted only to a laboratory. In gene therapy, vectors are used to carry the genes of interest into target cells. Both viral and non-viral approaches like microinjection, gene gun, etc. are used for gene delivery. Viruses act as a very specific and efficient gene carrier systems for both ex vivo and in vivo gene therapy modules. There are a number of viruses which are being used as vectors in gene therapy. The important ones include adenovirus, adeno-associated viruses, retroviruses, vaccinia virus, pox virus, herpes simplex virus, and others. Adenoviruses are most plausible candidates for gene therapy because of their high specificity and easy manipulation. They show efficient nuclear invasion and heightened expression. These viruses are especially suited for treatment of respiratory disorders because of their high affinity for respiratory cells. They can be used in the treatment of cystic fibrosis. However, some types of adenovirus may be cytotoxic with narrow host range. The other use of viruses is in cancer therapy. As you know, cancer is the uncontrolled growth of cells. Viruses can be directly used to prevent cancer by being a source of anti-cancer vaccines. For example, vaccines against hepatitis B virus. Hepatitis B virus, you might be knowing, is responsible for causing cancer called liver cancer or hepatic cancer. Human papillomavirus, responsible for treatment of cervical cancer, is also commercially available. During carcinogenesis, that is cancer formation, 6 fundamental properties are altered in the cells to give rise to destructive phenotypic cancer as illustrated in the antiviral vaccines. The vaccines are prophylactic in nature and stimulate immune system to synthesize and recruit specific antiviral molecules called interferons in cells. Cancerous cells express characteristic viral specific proteins. Thus, these could be targeted using vector to enhance specificity of vaccines. Such therapeutic vaccines are under clinical trials and are still not approved for human use. Virus-directed enzyme prodrug therapy, VDEPT, It involves uses of viruses to deliver an enzyme into a target cell. This enzyme can alter an inactive precursor of a cytotoxic drug into an active form. This treatment module is in research stages and is being evaluated for its therapeutic efficiency. Viruses are also useful in controlling harmful animals and insects. Some animals and insects which are harmful to human beings can be controlled by some specialized viruses which act as pathogens of these animals and plants. For example, T2 bacteriophage virus saves human beings from dysentery by spoiling some harmful bacteria like E. coli, Escherichia coli. Viruses are used in labs as the simplest living models. In the research of genetics, viruses are used. It is an important subject of genetic engineering. They help in identification of gene sequences, knowing presence of introns, exons, and help in transfer of genes from one cell to the other. Another role of viruses is in evolution. Viruses play a vital role to acquire knowledge about the trend of evolution and the process of formation of living organisms, because virus contains both living and non-living characteristics. This is because we place viruses at borderline between living and non-living organisms. Viruses are useful nowadays in paints and varnishes. High quality paints available nowadays contain viral particles which increase the shelf life of paints and protect timber, furniture from the attack of fungi, termites and other organisms. Viruses also find their use in textiles. High quality textiles used for making cloth are available in the market. The clothes have high polish and are 10 times durable in comparison to normal clothes. Viruses also find their use in wood polish. Polishing of wood is a common practice to develop natural grains to make wooden articles alluring. Wood polishes are available nowadays in market which contain viral particles that serve as antifungal, antibacterial and anti-insect agents. Thus, the polish retains its shine for longer period and the wooden articles are protected. Viruses also find their use in disease diagnosis. A number of viruses cause acute central nervous system disease. The two major clinical presentations are aseptic meningitis and the less common meningoencephalitis. Clinical virology laboratories are now more widely available than a decade ago. They can be operated on a modest scale and can be tailored to the needs of the patients they serve. most laboratories can provide diagnostic information on diseases caused by enteroviruses, herpes viruses, and human immunodeficiency virus. Antiviral therapy for herpes simplex virus is now available. By providing a rapid diagnostic test or isolation of the virus or both, the virology laboratories play a direct role in guiding antiviral therapy for patients with herpes simplex encephalitis. Although there is no specific drug available for the enterovirus, attention needs to be paid to the viruses since they are the most common cause of non-bacterial meningitis and the most common pathogens causing hospitalization for suspected sepsis in young infants in United States, especially during warm months of the year. When the virology laboratory maximizes the speed of viral detection or isolation, it can make a significant impact on the management of these patients. Early viral diagnosis benefits patients with enteroviral meningitis, most of whom are hospitalized and treated for bacterial sepsis or meningitis or both. These patients have the advantage of early withdrawal of antibiotics and intravenous therapy, early hospital discharge, and avoidance of the risk and costs of unnecessary tests and treatment. In diagnostic laboratories, virus infections are confirmed by several methods, which include: number one, growth of a virus in a cell culture. Number two, detection of virus-specific antibodies in the blood. Third, detection of virus antigens. Fourth, detection of virus nucleic acids. Gene sequencing to characterize virus strains. Next is observation of viral particles by electron microscopy. Another one is hemagglutinin assay. Viruses are also the agents that cause a number of diseases in plants. This leads to massive agronomic losses. Viruses cause a number of diseases in plants. However, there are limited studies in the field because it is difficult to cultivate and purify viruses. Some important plant diseases caused by plant viruses are tobacco mosaic diseases. It is caused by TMV. TMV is a thermal resistant virus which infects tobacco and other plants as well. It is transmitted through cell sap injury caused during clipping of the shoot. It has a positive sense RNA and codes for a protein like RNA polymerase, replication proteins, and movement proteins. The symptoms of TMV infection are mosaic and even blisters on leaves. Control of the disease can be carried out by crop rotation, sanitation, and use of resistant varieties. Another example is potato virus. It is present in potato plants worldwide and has a wide host range. The symptoms include mosaic, necrosis associated with dwarfing. Its genome is composed of a single-stranded positive sense RNA. This virus is transmitted through infected tubers and insects like aphids. Infection on crops with this virus causes huge economic loss. Another viral disease in plants is caused by cauliflower mosaic virus. This virus attacks cauliflower cells. It has double-stranded DNA as genetic material and infects a number of plant species besides cauliflower. This virus develops many symptoms which include leaf mottling and curling of edges. The fruits also show mottling with yellow and white patches. Cauliflower mosaic virus is transmitted by insects. This is all about the economic importance of viruses. Thank you.